Assalamu alaikum. Hope all of you are fine. This is our fifth lecture of the development of the nervous system. In this lecture, we'll discuss the development of the pituitary gland, the cerebral commissures, and few of the brain defects. The pituitary gland or the hypophysis cerebri is present in the hypophyseal fossa of the body of the sphenoid bone. The hypophyseal fossa is also called the sella turcica. The sella means saddle and turcica the Turkish. So it resembles the Turkish saddle. Therefore, it is called sella turcica. The pituitary gland is connected with the hypothalamus with the pituitary stalk or the infundibulum, and it has two parts: the anterior pituitary, which includes the pars tuberalis, pars distalis, and the pars intermedia, and the posterior pituitary part gland, which includes which is called the pars nervosa and the infundibular portion. The two portions, that is the anterior pituitary and the posterior pituitary gland, they have different origins developmentally. So we'll discuss the development of the pituitary gland. The pituitary gland or the hypophyseal cerebri is a combination of the two sources, that is the ectoderm and the develops from the ectoderm, which develops as an outgrowth from the roof of the stomodium. It is also called the Reckitt's pouch. It is formed during the third week of the development, and it grows upwards, whereas another diverticulum appears from the floor of the developing diencephalon from the neuroectoderm, and it is called the neurohypophyseal diverticulum. The two diverticula, they soon meet each other at the level of the infundibulum, and this is the neurohypophyseal diverticulum or the Reckitt's pouch is first in open communication with the oral ectoderm, and later on it is detached from this roof of the stomodium, and this forms the part of the site of the hypophyseal stalk. The two sources, the Reckitt's pouch or the hypophyseal diverticulum and the neurohypophyseal diverticulum, they meet, and the surrounding area which is developing in the body of the sphenoid bone is forming here. So later on, the hypophyseal fossa or the sella turcica is formed, and the pituitary gland from the two sources, they will meet each other and they will lodge in this hypophyseal fossa, and the surrounding area will form the sphenoid bone. So the neurohypophysis will form the pars nervosa of the pituitary gland, and this portion is from the infundibulum, whereas the hypophyseal diverticulum or the Reckitt's pouch will form the anterior pituitary gland, which includes the pars tuberalis, the pars distalis, and the pars intermedia. So the histologically also, the two sources of the pituitary gland, they can be identified as they have the different variety of the cells, and they are stained also differently. So the pituitary gland, which is a major endocrine gland, and it also controls the main hormones of the body. So it is developed from the amalgam of the two sources, that is the hypophyseal diverticulum and the neurohypophyseal diverticulum. This diagram showing you craniopharyngioma, which develops from the remnant of the hypophyseal diverticulum, and it is projecting towards the anterior part of the sphenoid bone. So this is a benign tumor, and it develops from the remnants of the hypophyseal diverticulum. Now, the development of the cerebral commissures. The cerebral commissures, they are the group of the fibers which connect the one part of the cerebral cortex with the another, or from one cerebral hemisphere to the other cerebral hemispheres. So these commissure fibers or the white matter of the cerebrum, it includes the lamina terminalis, optic chiasma, anterior commissure, then posterior commissure, and hippocampal commissure, then habenular commissure, and the corpus callosum. So these are all the cerebral commissures or the white matter of the cerebrum. So the lamina terminalis, it forms, this is showing you the lamina terminalis. So the lamina terminalis forms a bridge between the two hemispheres and enables the nerve fibers which passes from one cerebral hemisphere to the other. The remains of the lamina terminalis, which lie between the corpus callosum and the fornix, they will become stretched out and they will form a septum here, which is called the septum pellucidum. Now the anterior commissure. Now these anterior commissure fibers, they are first to develop and they run in the lamina terminalis and they connect the olfactory temporal lobes of the two sides. Whereas this hippocampal commissure, this hippocampal commissure fibers, they will form second to the anterior commissure, and these fibers, they arise in the hippocampus and they converge then on the lamina terminalis. Now, the most important commissure, which is the corpus callosum, and this is the third and the largest of the all the commissures or the white matter fibers or white matter of the cerebrum. It appears in the 10th week of the development and it connects the non-olfactory areas of the right and the left cortex. So, as a result of the continuous expansion of the neopallium, it extends anteriorly and then arch posteriorly. And over its roof is the lateral ventricle, whereas it forms the roof of the third ventricle. So this is diagram showing you the developing corpus callosum, and so you can see the forms the head, body, and the tail, and it over it lies the lateral ventricle, and here the third ventricle will form in which and the corpus callosum will form its roof. Now the optic chiasma, it is formed at the inferior part of this lamina terminalis, and optic chiasma will contain the fibers from the medial halves of the retinae. So the fibers, they cross the midline to join the optic tract of the opposite side. Now you can see all these structures. So this is the cerebral cortex, the corpus callosum, the septum pellucidum, and this is the thalamus and the hypothalamus, interthalamic adhesion, and this area is the lamina terminalis and the optic chiasma, the pituitary gland, and this is the mammillary body, whereas this is the pineal gland. Now this is showing you a diagram showing you the relationship of all these commissural fibers. You can see here the corpus callosum, the septum pellucidum, and this is the fornix and the hippocampal commissure. So all these commissural fibers, they are in relation with each other. Now, these certain brain defects. So you should know the abnormal formation of these brain defects. What is the abnormality, and then discuss these brain defects. For example, this is the diagram showing you the different varieties of the encephalocele. An encephalocele results from the non-closure of the anterior neuropore. So as a result, there is the herniation of the brain tissue outside. With or without the brain tissue, there is an herniation, and there are different varieties. For example, this is showing you the defect at the level of the foramen magnum, and in which the parts of the cerebellum, they are also protruding out. whereas there is a defect if they at the level of the posterior fontanelle, and the meninges they are protruding out, and there is a dilated portion, whereas this is the abnormal formation of the occipital lobe. And this diagram showing you the defect at the level of the posterior fontanelle, and the parts of the occipital lobe they are protruding in this area. This scan or CT scan is showing you the heterotopic cerebral cortex. The heterotopic cerebral cortex is due to the abnormal migration of the neuroblast cells. So this is called the heterotopic cerebral cortex. This is the common asked question about in the MCQs also about the hydrocephalus. The hydrocephalus results in the enlargement of the head, and this diagram showing you a hydrocephalus baby with the cleft lip also. So there is an increased amount of the CSF which flows in the ventricles, which results in the dilatation of these ventricles, and it is due to the imbalance between the formation and the absorption of the cerebrospinal fluid. So a lot of CSF it is present in the ventricles, which results in its dilatation and the enlargement of the head. This is the diagram showing you the brain with the holoprosencephaly. In the holoprosencephaly, the midline structures they do not detach, and there is a defect in their development. This is showing you the developing telencephalon, and these are the cerebral hemispheres, developing cerebral hemispheres. So they will not detached from the midline, or the right and the left hemispheres they are not formed, and if there is a single hemisphere is formed. So this is called the holoprosencephaly, where is a defect in the midline structures. This is a diagram of the MRI scan showing you the Arnold-Chiari malformation. In this malformation, there is the defect in the there is a herniation of the parts of the brain through the foramen magnum, and this is showing you the tongue-like shape of the medulla and the herniation of the cerebellum and into the foramen magnum. So this is a brief summary of the all the anomalies of the brain. So anencephaly is the defective formation of the skull with exposed malformed brain. Hydrocephalus is the distension of the ventricles due to the accumulation of the CSF. Microcephaly is the small sized brain and the skull. Meningocele is the protrusion of the meninges through the skull defects. Meningoencephalocele is the protrusion of the meninges along with the brain tissue. And then another variety is the meningohydroencephalocele, where is the protrusion of the meninges, brain tissue, and there is also a dilated ventricles. Then holoprosencephaly is the malformations of the midline structures of the head in the brain and the face. Then schizocephaly is the large cleft in the cerebral hemisphere. Then Arnold-Chiari malformation is the caudal herniation of the cerebrum through the foramen magnum. So these is the brief summary of all the brain defects, and you should know the major abnormality causing these brain defects. So this is the our last lecture of the development of the nervous system. So we have discussed all the structures in detail. So if you have any queries, you can ask me from the WhatsApp group, and give me the feedback of all the lectures about the development of the nervous system. Thank you.